Yes, we live now. Welcome to Let's Learn Bio by NCA. This is Nirupadesh Amdiyal. In this session, we are going to discuss biotechnology principle and process. Within that, the first concept, introduction, and principles of biotechnology. First question: which of the following is an incorrect statement? Eco R1 was first restriction enzyme to be discovered. This is incorrect. Here, restriction enzymes are obtained from bacteria. Correct statement. Restriction endonucleases are called molecular scissors. Correct. Antibiotics, vaccines, enzymes, etc. are all biotechnological products. Correct. Incorrect. Third statement. Third option. The role of DNA ligase in construction of recombinant DNA molecule is the role of DNA ligase is the formation of phosphodiester bond between two DNA fragments. The formation of glycosidic bond takes place between monosaccharide units leads to formation of disaccharides, oligosaccharides, polysaccharides. Cutting of DNA at specific site, it is the role of restriction enzymes. The fourth option is the ligase action, role of ligase. Third question, the integration of natural science and organism, cells, parts thereof, and molecular analogues for products and service. This definition of biotechnology was given by European Federation of Biotechnology, EFB. Third option is correct. GEAC, Genetic Engineering Approval Committee. This is it take it make a decision about validity of GM research and safety of introduction of genetically modified organisms for public service. CDRI, Central Drug Research Institute, Lucknow, present in Lucknow, it developed Shaheli, the oral contraceptive. The fourth option, International Union of Biotechnology, it is the EF definition given by this European Federation of Biotechnology. In genetic engineering, recombinant DNA means DNA with a piece of RNA, DNA which take part in recombination, DNA with a piece of foreign DNA, DNA not associated with the recombination. Third option, DNA with a piece of foreign DNA is the recombinant DNA. Next, fifth question, gene cloning means. Gene cloning means producing a new genotype, producing genetically modified organisms, obtaining identical copies of particular DNA molecule, and preserving a genotype, replacing mutated gene with normal gene. Gene cloning means obtaining identical copies of particular DNA molecule and preserving the genotype. Third option is correct. Sixth, given below are two statements. One is labeled as assertion A and other is labeled as a reason. First, assertion. When a piece of DNA get integrated into the genome of a recipient, it may multiply and be inherited along with the host DNA. Reason, the alien piece of DNA has become a part of a chromosome which has ability to replicate. In the light of above statements, choose the most appropriate answer from the option given below. Here, both assertion and reason are correct. Reason R is correct explanation of A. The fourth option is correct. Next, given below are two statements. Statement 1, making curd, bread, wine, which are all micro-mediated process, could also be thought as a form of biotechnology. Correct statement. Biotechnology is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Again, correct. So, both statement 1 and statement 2 both are correct. The enzyme used to combine two fragments of DNA is DNA ligase. It's also referred as molecular glue. So, option 2, 1 and 2, option 4 is correct. Next, ninth, identify the correct set of statements. Plasmids, bacteriophage, and retroviruses can be used as a vector to deliver an alien piece of DNA into the host organism. Correct. First artificial recombinant DNA molecule was constructed in year 1972. Correct. In a chromosome, a specific DNA sequence which is responsible for initiating replication is called origin of replication. Correct. A sexual reproduction preserves the genetic information while the sexual reproduction permits variation. Again, this is also correct. So, option 4, A, B, C, D all are correct. Next, match the following columns and choose correct option. Column 1, René Descartes, the biologist of 17th century. Cohen developed a method of removing plasmids from a cell and then reinserting them in the other cells. Boyer stated that restriction enzymes have capability of cutting DNA strands in a particular fashion. So the first option is correct. Next, what are the basic steps in genetically modifying an organism? Identification of DNA with desirable genes. First, second, introduction of identified DNA into the host, and maintenance of introduced DNA in the host and transfer of DNA to its progeny. That is a second option. First, third, and fifth statements are correct. Next, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in a host organism. This can also be called as the process called cloning. Next, the technique of genetic engineering includes. The technique of genetic engineering includes creation of rDNA, gene transfer, and gene cloning. Three processes together. All of the above is correct. When a recombinant DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate and make multiple copies by using the DNA polymerase enzyme of a host cell. DNA ligase enzyme joining the DNA fragments. Liases breaking down. Reverse transcriptase. These enzymes catalyze the formation of DNA from RNA as a template present in retrovirus. Next, the construction of first recombinant DNA emerged from the possibility of linking a gene encoding the antibiotic resistance with the native plasmid of Salmonella typhimurium. Agrobacterium tumefaciens, it is a natural genetic engineer. It is used as a vector for transfer of gene into the plant cells. Bacteriophages, they are used as a vector to transfer gene into bacteria. Escherichia coli, they are bacteria, commonly using bacteria. The disadvantage of traditional hybridization procedure used in plant and animal breeding is the traditional hybridization procedures, it inclusion and multiplication of undesirable genes along with the desired genes. This is a main disadvantage of conventional or traditional hybridization procedure. Yes. Next, tools of recombinant DNA technology. Identify the wrong statement with respect to restriction enzymes. More than 230 restriction enzymes have been isolated from 900 strains of bacteria. This is a wrong statement. 900 restriction enzymes have been isolated from over 230 strains of bacteria. Next, the specific DNA sequence where EcoR1 cut is. The restriction specific recognition sequence of EcoR1, that is GAATTC. Next, which of the following is not a vectorless method of gene transfer? That is Agrobacterium tumefaciens. An ideal cloning vector should possess restriction sites, selectable marker, origin of replication, all of the above. Fifth, which of the following is correct with respect to movement of DNA fragments on agar gel during gel electrophoresis? Negatively charged DNA fragments do not move. Negatively charged molecules move from anode towards negative towards a positive side, that is towards the anode. The smaller the fragment size, farther it moves. So this is a correct statement. Positively charged DNA fragments move towards a farther end. No, negatively charged DNA fragments move farther end. Larger the fragment size, farther it moves. This is wrong. Larger the fragment size, near its moves, very near to the well, not farther. Next, sixth, in genetic engineering, the probe refers to, it is a radioactively labeled single stranded DNA is called probe. Next, which of the following is an incorrect statement? DNA fragments are negatively charged. Correct. A gene whose expression helps to identify transformed cells is known as selectable marker. Correct statement. Exonuclease enzyme cut the DNA within the specific position. This is an incorrect, wrong statement. Exonucleases cut or remove the nucleotides from their free ends, not within a specific position. Bacteria protect themselves from viruses by fragmenting viral DNA upon entry with the endonucleases. This is correct. So incorrect statement, third one. Which of the following is incorrect with respect to plasmids? It is a circular double-stranded DNA, correct. It carries antibiotic resistance gene, correct. It has ability to autonomous replication, yes, correct. It is under the control of chromosomal DNA, this is incorrect. Fourth one is incorrect. Next, given below are two statements. One is labeled as assertion and other is labeled as a reason. Assertion A, assertion, the DNA fragments can be separated by forcing them to move towards anode under an electric field through medium or matrix. Yes, correct. Reason, DNA fragments are negatively charged molecules. Here, assertion and reason both are correct. Again, reason is a correct explanation for A. Next, restriction endonucleases are utilized in genetic engineering as restriction endonucleases are utilized as molecular cement for combining DNA fragments into long. This is molecular cement means DNA ligases, molecular glues, not endonucleases. These are the tools of recombinant DNA technology, correct. Molecular scalpels for cutting DNA at specific site, correct. So here second and third both statements are correct. Fourth option is correct. Next, in order to induce bacterial uptake of plasmids, the bacteria are made competent by first treating them with calcium chloride, bivalent cation. Next question, find out the statement that does not hold true for restriction enzymes. Does not hold true. It produces the same kind of sticky ends in a different DNA molecules, correct. It is an endonuclease, yes. It is isolated from virus. Restriction enzymes are not isolated from virus. They are from bacteria, prokaryotes. This is incorrect, not hold true. It recognizes palindromic nucleotide, correct. For this question, does not hold true for restriction enzyme. Third option is. Next, in 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. These enzymes are first enzyme methylase, second restriction endonucleases. Methylase add methyl group to the host DNA, thereby it modify their DNA and it protect the host DNA. Whereas restriction endonucleases, non-methylated DNA, it is recognized them as a non-self, then it is subjected for cleaving. For next question, which of the following statement is incorrect about gel electrophoresis? Incorrect. The most commonly used matrix agarose, yes, correct. DNA fragments move towards cathode under electric field. No, the DNA fragments move towards anode, not towards cathode. This is incorrect. DNA fragments are separated according to their size under electric field. Yes, this is correct. Bright orange bands are seen after staining the DNA with ethidium bromide. This is a stain used to visualize separated DNA bands on autoradiography. Next, which of the following describe the role of selectable marker? The selectable marker helps in differentiating between transformants and non-transformants. The site where replication start, it is called ORI site, origin of replication. The vector used to selectively for plants, that is Agrobacterium tumefaciens. It is a cloning site for alien DNA. Cloning sites are different sites: BAMH1, SAL1, CLA, HIND3, HIND2, EcoR1, SAL, PST1, PVU1. These all are the cloning sites. Selectable marker helps to differentiate transformants from non-transformant. Next, which of the following is a incorrect statement regarding enzyme used for recombinant DNA technology? Incorrect statement. DNA polymerase remove phosphate group from 5' end of double stranded DNA and prevent unwanted ligation. This is incorrect. DNA ligase help in sealing a gap, act as a molecular glue, correct. Reverse transcriptase is used to form DNA from mRNA. It is called complementary DNA or cDNA, correct. Third one is incorrect. Next, identify the wrong statement. DNA is a hydrophilic, not hydrophobic molecule. This is a wrong statement. DNA cannot pass through the cell membrane, correct. Treatment with the divalent cation increases efficiency with which DNA enter the bacterium through pores in its cell wall, correct. Disarmed pathogen vectors, when allowed to infect the cells, transfer RDN into the host, correct. Next, during the formation of recombinant DNA, the plasmid vector is cleaved by. Plasmid vector is cleaved by the same enzyme that cleaves the donor DNA. Both, so donor as well as vector plasmid DNA are treated with the same restriction enzymes. Next, the colonies of recombinant bacteria appear white in contrast to the blue colonies of non-recombinant bacteria because the insertional inactivation of beta galactosidase in recombinant bacteria takes place. This is correct first. Next question. Match the following columns. Column 1, column 2. Insertional inactivation beta galactosidase. Recombinant protein heterologous host. Palindrome Malayalam. Recognition sequence GAA-TTC. The term palindrome means the group of letter forms the same word from forward as well as from backward. Palindrome. Malayalam, Gadag, etc. The third option is correct. Next, given below are two statements. Assertion reason. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure. Yes, correct statement. It requires simultaneous plating on two plates having different antibiotics. Yes, correct. Assertion, reason, both are correct, and the reason is explanation for A. Next, the process of recombinant DNA technology. The application of a PCR technique is the PCR. It helps to make multiple copies of a specific DNA sequence. It is called amplification of nucleic acid. Second option. PCR proceeds in three distinct steps governed by temperature in the order. That is first denaturation, annealing, then extension. 3 sequential steps of a PCR. Second option. Next, read the following statements about downstream processing and choose a correct option. Downstream processing and quality control testing vary from product to product. Correct. The product has to be formulated with suitable preservatives. Correct. Strict quality control testing for each product is required. All of the above 3 statements are correct. Next, the utilization of Taq polymerase in PCR serves the purpose of Taq polymerase. It fast amplification of sample of DNA at a very high temperature. Second option. Taq refers to Thermus aquaticus. It is extracted from bacteria. Thermus aquaticus. It is most thermophilic. Next question. Match the column. PCR amplification of gene of interest, gel electrophoresis, separation of DNA fragments, downstream processing, separation and purification, bioreactors, culturing host cells. So the third option is correct. Next, the PCR technique is used for PCR. It is the in vitro synthesis of specific DNA sequences using thermostable DNA polymerase. Just told Taq polymerase, it is a most thermostable DNA polymerase. It is active even at high temperature. Third option. During the process of isolation of DNA, chilled ethanol is added to precipitate purified DNA. First option. Break open the cell to release DNA. Different enzymes are treated like cellulase, plant cell treatment, chitinase to break fungal cell wall, lysozyme to break open bacterial cell wall. Remove proteins. Proteins were removed by using enzyme protease treatment. Facilitate action of restriction enzyme. Restriction enzymes only cut the DNA. The DNA should be in a pure form. So the first option is correct. Given below statements assertion and a reason. Assertion: restriction enzyme digestion are performed by incubating purified DNA molecule with restriction enzyme at the optimal condition for that specific enzyme. Correct. The purified DNA can be seen as a collection of fine threads in suspension. Correct. But reason is not correct explanation for A. The first option is correct. Next, the process of separation and purification of expressed protein before marketing. This is called downstream processing. Next, bioreactor has an agitator system, an oxygen delivery system, a foam control system, temperature control system, pH control, and a sampling port. Yes, all of the above. Sorry, bioreactor as a second, third, fourth, fifth, and a sixth. Fourth option is correct for this. Agitator system is present in sewage treatment. A recombinant DNA was generated using human gene. It was used to transform a bacterial cell, yet the desired protein was not produced by transformed cells. What could be the reason behind all of this? First, human proteins formed are degraded by bacteria. Amino acid codons for humans and bacteria are different. Human genes may have introns which bacteria cannot process. Yes, this is third option is correct. Read the following statements and choose the incorrect option. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant or animal cell, alien DNA get multiplied. Correct. The cells can also be multiplied in a continuous culture system wherein used medium is drained out from one side while fresh medium is added from other side to maintain cell in their psychological. This is wrong. In their most physiologically most active or log exponential phase, not a psychological. Second option, this is incorrect. After the completion of biosynthetic stage, the product has to be subjected through a series of process before it is ready for marketing as a finished product. This is a downstream processing, correct. Second option is incorrect. Elution means, elution it is the cutting and extraction of DNA bands from agarose gel. This is a elution. The transfer of DNA on nitrocellulose membrane, this is called blotting. Binding of a probe with a specific sequence called hybridization. This is isolation from a chosen organism. Normal, that is an isolation process. Next, bioreactors can be thought of as vessels in which the raw materials are biologically converted into specific product. The first option is correct. Next, if a protein encoding gene is expressed in a heterologous host, it is called recombinant protein. Simple protein consisting only the one type of protein, amino acids. Simple protein. Conjugated protein, different subunits of protein interacted, conjugated protein. Denatured protein means protein that lost their different structural levels. It exists only in primary form, denatured. Next question. Dash has resulted in the popularity of PCR, polymerase chain reaction. The availability of thermostable DNA polymerase is the popularity of PCR. First option. Next, match the column 1 and column 2. Thermostable DNA polymerase, that is a Taq polymerase. Joining of the DNA molecules, that is a DNA ligases. Large scale production of products, bioreactors. Recombinant protein, heterologous host. So the third option is correct. The last question. For the production of a biotechnological product at manufacturing scale, bioreactors are used for which all statements are correct except the stirrer in a tank. Bioreactors help to mix the reactor content and facilitates oxygen availability throughout the bioreactor. Correct. Sampling port in bioreactors are meant for adding raw material into them. This is wrong. Sampling port means small amount of product is periodically removed, not added. Second is incorrect. Optimum conditions such as pH, temperature, etc. for production of biotechnological products are monitored in them. This is correct. Air bubbles bubbled through the reactor dramatically increase oxygen transfer area. Correct. This is exceptional case. Second one.